Hi, welcome back everyone. Let's continue our discussion of different effects of beneficial and deleterious mutations and the underlying statistical property associated with them called distribution of fitness effects. To understand this, let us consider a genome. So, this is a genome of an organism. This could be E. coli, maybe we have linearized the circular genome, but this is the finite piece of genome of any organism. And the experiment that we will do is that in this genome we will introduce one mutation, and let us imagine that that mutation was a deleterious mutation. So, I will indicate all deleterious mutations with red and all beneficial ones with green. So, this was a deleterious mutation whose selection coefficient was SD1, selection coefficient of deleterious mutation number 1. After this, let me just draw these bunch of genomes. After this, I introduce beneficial mutation. I introduce a mutation in the original genome once again, and let us imagine that this mutation also was deleterious. So, this was also deleterious and its selection coefficient was SD2. I go back to the original genome and introduce another mutation randomly, and this time the mutation was beneficial. This was a beneficial mutation and its selection coefficient is SB1, and so on and so forth. I introduce a mutation in the ancestral genome, measure its fitness effect, and then go back to the ancestral genome and introduce another mutation. At the end of this exercise, what I am going to have here, so let us say I did lots of, let us say I did this exercise hundreds of times. At the end of these hundreds of times, let us say I will get some deleterious ones, but I will also get many beneficial ones. So, the first thing I will do, so this, all of this happened. First thing now we will do is understand that we will make a list of the fitness effects of all beneficial and deleterious mutations. So, this is, we are going to make a table which is beneficial mutations and deleterious mutations, and we are going to list the benefit or the deleterious effect conferred by each one of them. So, we will start with the first one. This was a deleterious mutation whose effect was SD1. So, I write SD1. Next one was also deleterious, its effect was SD2. So, I write SD2. The third one is beneficial and its effect was SB1. So, I write it in the column of SB1, and so on and so forth. Let us say this was SB2, SD3, and so on and so forth. So, I write down this table and I have a whole bunch of data points here. So, let us say I have SDN and SBN data points in this table. These are individual values of the selection coefficient associated with beneficial or deleterious mutations that are randomly introduced in a genome that I wanted to study. Let us imagine that the number of mutations that I studied was 100 in each case. So, n is equal to 100. I studied 100 beneficial mutations and I studied 100 deleterious mutations. Let's only worry about the beneficial mutations when we are trying to understand this. We will make the following plot. On x axis is S and on y axis is the percentage of mutations. So, we ask that how many mutations confer a benefit which is between 0 and 0.01. Remember last time we studied, we made up these two examples where the selection coefficient of the first beneficial mutation was 0.05 and selection coefficient of the second beneficial mutation was 0.03. But there will also be some mutations whose selection coefficient is, let us say, 0.006. So, that one will be counted in this particular window. So, among these 100 we count how many have a selection coefficient which is between these two numbers, and that, let us say, the number is 15 out of 100. So, 15% mutations confer benefit between 0.001 and 0 and 0.001. Next we ask how many of these mutations confer a benefit between 0.01 and 0.02. So, maybe there is a beneficial mutation whose selection coefficient is 0.015. So, that gets counted, and maybe there are 30 such mutations which confer benefit or selection coefficient between 0.01 and 0.02. So, that is 30. That is 30 plus 15, 45 done. Let us say in the next window there are 0.03. How many are between 0.02 and 0.03, and maybe the number is 25. That is 40, 70 and points. We continue this 0.03 and 0.04, maybe the number is 10, that is 80, and then another 10, 0.05, that is 90, and then it will be not many left, and maybe there was just one mutation that conferred this really high benefit. This distribution tells us that this tells us two things. One is that not all beneficial mutations are same. There are some mutations which confer a very large benefit, the value of their selection coefficient is very high, but there are many mutations who confer a very small benefit, the value of their selection coefficient is really small, and in between there is a distribution. So, first this experiment confirms that not all beneficial mutations are same, and second, the distribution of benefits, the distribution of benefit that a mutation confers is like the figure that I just saw. So, it gives a quantitative description of the first statement. The first statement is simply saying that they are not same, they are different, but it does not tell me that what does the distribution look like. This distribution is useful because it tells me that more often than not, when a beneficial mutation occurs, chances are that its benefit will be within this region. Beyond that, beneficial mutations are available, but the benefit beyond that beneficial mutations are available, like this one is a great beneficial mutation which confers the highest selection coefficient in all the beneficial mutations I tested. However, their frequency, the percentage with which they occur is very small. So, this tells me that more often than not beneficial mutations of small selection coefficients will occur, and it will only be once in a while, rarely, that a beneficial mutation with a very large selection coefficient will take place in the population that I am studying. And the same exercise can be done for deleterious mutations. This distribution is called distribution of fitness effects. Later on in the course, we will discuss a very recent paper which tries to measure this and does measure what is simply called DFE, distribution of fitness effects. So, what do we know so far about from the, so this was a conceptual discussion of what a DFE is, but from experiments, what do we know about DFEs? We know the following. This is for beneficial, and this is for deleterious. For beneficial, what we know from experiments is that this distribution looks like the following, that there are lots of mutations which confer, and obviously this is S and this is percentage of mutations. There are lots of mutations which confer small benefit, but as we are moving towards higher values of S, the mutations decrease rapidly. So, this distribution of beneficial mutation tells us that beneficial mutations obey this statistical law, which says that there are lots of mutations with small effects, with small benefit measured as S, but there are very few mutations with large effects. That is the general consistent theme that we have learnt about distribution of fitness effects through many experiments in many different species of organisms that this has been studied in. Deleterious mutations we know comparatively less as compared to beneficial mutations, because deleterious mutations are anyway going to be weeded out from the population, because a deleterious mutation happens and natural selection gets rid of that individual. So, they have been paid less attention, but what we do know, and obviously in deleterious mutation S is going to be negative, this is 0, this region is only beneficial mutations, and that is the graph here, and this region, this is 0, this is positive S, and this region here is all deleterious mutations. So, and this is again percentage of mutations, and what we know is that this distribution also follows a similar statistical pattern, but slightly more broader. Something like this. So, this here is the DFE of beneficial mutations, and this is the DFE of deleterious mutations. And as I said that this will have very interesting implications that we will study in a few lectures time. Right. So, that is the distribution of, that is our discussion on distribution of fitness effects. Moving forward, we want to discuss a geometric structure which was proposed almost 100 years ago, which is extremely useful in order to view evolution. In this geometric structure, we are going to, the analogy that we are going to use is that of a mountain climber. If you imagine yourself climbing a mountain, what you are seeing here is that if this is the mountain and then you are climbing it up, your path is rarely going to be straight up. The path is going to be a meandering up that you take, eventually leading to summit of the mountain. And in this analogy that was proposed in 1932, evolution is viewed in much the same way as one climbing up the mountain. The idea being that the height of the mountain, this axis, is the fitness of the population. And as a starting population, you are here. So, the starting population is at a very low fitness. In this axis, the value is very low. But then eventually, one individual acquires a mutation and moves to a slightly higher fitness value. When this slight movement to slightly higher fitness value happens, the progeny of this more fit individual are able to outcompete the progeny of the ancestors' children. Hence, the entire population shifts to a higher fitness level. And now the population is at somewhat higher level. And then again, another beneficial mutation will take place in one of the individual's progeny, which will then lead to that progeny's offsprings outcompeting the ancestral population and another step up, and so on and so forth. So, evolution can be viewed as a process which is sort of climbing this mountain. And this geometric structure or this geometric view of viewing evolutionary processes, this structure is called a fitness landscape. So, we are going to study conceptually in the next 1 or 2 videos conceptually what are fitness landscapes, and after that we will discuss 3 examples of what do we actually know about fitness landscape structures from real experiments. We will discuss 3 such examples, one from a paper in 2005, another one in 2013, and the last one which was published last year. Okay. So, this begins our discussion, which will continue for a few lectures on these structures called fitness landscape. And in its most general sense, the idea is as follows, that here you have a mountain climber which is going up. So, this mountain climber is going up the peak, and on the left side I will draw the parallel associated with an evolutionary process. That imagine that a population is here. So, this axis here is fitness in the case of evolution and height of the mountain in the case of mountain climber. So, imagine you have a population which is at a very low fitness. Now, this population is dividing, and if there is no mutation happening, then it really is not changing in its fitness level. But at some point a new individual is going to come up which is fitter than the rest, and as a result this individual is going to produce more progeny and it will outcompete the ancestral population, and as a result the population moved from its previous level to this new level, and this has resulted in this gain of fitness, just as climbing up the mountain leads to gain of height. But this process will not stop here. This will continue till one of the individuals here picks up another beneficial mutation, and then the progeny of the blue individual will outcompete the progeny of the green individual because it is fitter, and hence the population moves here, and in the process the green population is lost. This has resulted in gain of fitness once again, and this process continues as the population climbs this mountain and eventually reaches what is described as the fitness optima of a population. So, this is the analogy that is used while viewing evolution as a mountain climbing sort of a process. So, the analogy that this draws is between mountain climbing and evolution. So, this is the geometric view of evolution, which is an extremely useful analogy. It gives us a physical structure in which to view an evolutionary processes that we will be spending the next few videos on. This was first proposed by an American geneticist called Sewall Wright in 1932. So, in a few years time it is going to be 100 years when this was first proposed. All right. So, when as you might know, that the structure of DNA was published in 1953. The structure of fitness landscapes, the idea of fitness landscapes was proposed, as we just saw, in 1932. So, more than 20 years before we even knew the structure of DNA. As a result, the interpretation of fitness landscape and the population context in which Wright proposed is slightly different from what we are going to use. So, the way I am going to describe fitness landscape in these videos is going to be from a molecular perspective, when we know the DNA and all the important macromolecules inside a cell. And also Wright's proposal of landscapes was from the context of sexually reproducing populations, because work on microbes was not really that prevalent back then, but our description is going to be from a molecular perspective of DNA, RNA or protein. So, how do we think about this? The way we need to define a few concepts. The first thing we want to define is something called sequence space. We will imagine a linear sequence of DNA, whose length is L. L is the length, which is simply the number of nucleotides in this sequence. So, the question we need to first ask is how many unique DNA sequences are possible each of them being of length L. So, we ask that how many DNA sequences are possible, each of which is of length L. And this is not really that hard. Let us imagine that L is 1. So, we are interested in DNA sequences whose length is only 1, which means there is only one nucleotide in the DNA sequence. What you should realize here, that there are 4 such possible sequences. For DNA sequence of length 1, it could be, we saw that DNA is directional. So, 5 prime A 3 prime, 5 prime T 3 prime, 5 prime G 3 prime, 5 prime C 3 prime. This is simply written as A T G C. So, you could pick out any one of these 4 nucleotides that we know make up DNA, and that gives you a unique DNA sequence. Hence, the answer to this question for L equal to 1 is simply 4. Suppose L was equal to 2, in that case how many such unique DNA sequences can we make? And now we have to fill these 2 places. So, for the first place we have 4 choices. We can pick A, T, G or C, and for the second position we again have 4 choices, A, T, G, C. Because if we pick an A here, picking an A again is allowed, because repeats are allowed. Remember, our nucleotide sequences are billions of bases long, but they are only comprised of 4 bases, these same ATGC. So, there are 4 choices here and there are 4 choices here. So, the total number of sequences that you could build is 16. One way to think of this could be that this is the first nucleotide, this is ATGC, it could be any one of the 4, and this is the second nucleotide, which is ATGC. And now to make a sequence, you simply pick one from the first position and another from the sequence. So, this intersection of these two gives me the sequence as AT. This is AA, AG, AC, and similarly TA, TT, TG, TC, GA, GT, GG, GC, CA, CT, CG, and CC. So, there are again these 16 possible sequences if the number of length, if the length of the DNA fragment that I am looking at is equal to 2. Similarly, if for any general sequence of length L, the number of sequences that is available, that is possible, is simply equal to 4 to the power L. And we just did the cases of L equal to 1 and L equal to 2. So, that is the number of possible DNA sequences of length L, and as you should realize that when we are talking of genomes of an organism, even something as simple as E. coli, whose genome is going to be 5 into 10 power 6, even for that the number of possible DNA sequences of length equal to E. coli's genome is equal to 4 to the power 5 into 10 power 6. This is an astronomical number, much greater than the number of atoms that is present in the universe. This is the number of possibilities that exist of building DNA sequences of length L. All right. So, we have for length L, we have 4 to the power L possible sequences. Which is a huge number if L is genome of an organism or even a gene. What we are going to do in building this geometric construct is that we will represent each sequence, each of these 4 to the power L sequences with a node. So, I have 4 to the power L possible sequences and each sequence is represented by a node. So, this is sequence number 1, this is sequence number 2, sequence 3, sequence I, and so on and so forth, eventually ending at sequence number 4 to the power L. From the context of the 2-node experiment, 2-node case that we discussed on the previous slide, this is going to be AA will be one node, AT, AG, AC, TA, TT, TG, TC, GA, GT, GG, GC, and CA, CT, CG, and CC. That is what we are aiming to make here. And as you can see that even with L equal to 2, writing this was a bit challenging, but as L increases, this is going to get astronomically big and handling this manually is going to be impossible. This set of all possible sequences of length L is called sequence space associated with this length. So, a sequence space of L equal to 2 consists of 16 nodes, whereas sequence space associated with a length of DNA equal to L consists of 4 to the power L nodes. And obviously, this is a very large number. So, that is the idea of sequence space. So, where we are at is that we have this sequence space of size 4 to the power L, where each of these sequences is represented by one node. Next, what we will do in this is that we will define a quantity called Hamming distance between these sequences. Hamming distance is defined by definition, it is only defined between two sequences. So, we do not define Hamming distance between 3 or 4 sequences, it is done in a pairwise manner, where of the set of sequences that we have, we take 2 and then we define the Hamming distance between them. It is only defined between 2. And the second thing is, Hamming distance is only defined if sequences being compared, let us call the two sequences that are being compared as sequence I and sequence j. It is only defined if sequence I and j are of same length. So, in the previous example that we looked at, all of these 16 sequences are of length 2. So, I can pick any one of the 2 and define the Hamming distance between them. However, what I cannot do is define a Hamming distance between one sequence from this group and one sequence from this group, because these are nodes which represent sequences of length L, whereas these are nodes which represent sequences of length 2. So, Hamming distance is only defined in a pairwise manner and only between sequences of equal length. So, given sequences I and j, Hamming distance, it is represented as Hij. Hamming distance between sequences I and j is equal to number of nucleotides where the two sequences are not identical. What does that mean? So, if I am comparing, if my sequence I is AA and sequence j is AT, then Hij is simply equal to, at what position are these two sequences not the same? At the first position they are identical, but at the second position they are not the same. So, Hij is simply equal to 1. Compare another sequence I, which is a sequence G, G, C, C, A, A, T, T, and another sequence J, which is G, 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 A, 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 A. In this case, Hij is simply equal to 1, 2, 3, 4, the 4 places where these two sequences are dissimilar from each other. So, Hij in that case is 4, Hij in this case is 1. Of course, I can also have cases where I is equal to AA, J is equal to AA, and in that case Hij is simply equal to 0. That is the definition of Hij, and we will continue to build these fitness landscapes using the sequence space that we just built on this slide and using the idea of Hamming distance and work towards developing this theme.